Hello and welcome everyone. In this podcast, we're going to begin our discussion of transport across a membrane. So let's begin by drawing a cell to introduce the problem here. Up to this point, we've talked about membranes, as I'm drawing here this plasma membrane. We've talked about reactions in a cell caused by enzymes. We talked about various organelles in the cytosol here, the nucleus, the mitochondria, and we'll come back to these later on, but we did mention them, and some others. We also talked about, within these membranes, specific proteins that could be on the outside, could be on the inside. They might go through the membrane. We talked about the membrane itself, how within the membrane we had a hydrophobic portion, and on the outside of the membrane we had a hydrophilic portion. We talked about how these lipids here could have sugars attached to them or a phosphate group. But we haven't really addressed how this cell is going to survive. Cells don't exist in a vacuum. They exist within an environment. They have to have some way to exchange molecules with the environment. Potassium, sodium, calcium, oxygen, CO2, glucose. These all are outside the cell, and they're also all inside the cell. So there has to be a mechanism to send these ions back and forth to maintain a proper homeostasis within the cell. Some of these will be easier to move across the membrane. Some of them will require more energy, more work. But it's important that the cell has a way to exchange with the environment these particular ions and other ions. So that goes hand in hand with this second one, and that's regulating the concentration of ions in the cytosol and organelles. So proper concentration of these ions has to be maintained on the outside of the cell, in the cytosol, and within these various organelles. The next one here is import nutrients. And I already wrote that here with glucose, but it could also be amino acids that have to move into the cell. So there has to be a mechanism to bring in nutrients into the cell. Likewise, we have to export waste from the cell. These various enzymatic reactions generate some sort of waste product, and those waste products have to be removed from the cell in order for the cell to survive. So over here I mentioned at the very beginning is that we have to have a mechanism to exchange ions across the membrane. Sodium, potassium, calcium, oxygen, carbon dioxide, and other ions. So let's consider the type of solute in order to understand whether or not it will easily cross the membrane or if it will require an investment of energy to do so. So let's list some of these factors that affect movement across a membrane. Okay, the first one is size. That might not seem too surprising, but we know that smaller molecules diffuse easier than larger molecules. The second factor is polarity. Nonpolar molecules diffuse easier compared to polar molecules. And the last one is whether or not the molecule is ionic. Does it have a charge? And that's because the membrane is very impermeable to ions. So let's over here draw a membrane, and let's start with small hydrophobic molecules. This would be like oxygen, CO2, If we look at our rules over here, it's small and it's hydrophobic, nonpolar. So these will easily diffuse across the membrane. Next, let's consider small, uncharged polar molecules. This would be like water and ethanol. These will also diffuse across the membrane. Next, let's look at larger, uncharged polar molecules. This would be like glucose, nucleotides, and amino acids. These are not going to easily move across the membrane. They can still get across the membrane, and we'll talk about that in a moment, but they're going to require some extra help getting across the membrane. And lastly, let's consider our ions. These could be sodium, potassium, calcium, chloride, and lots of others. And even though these are very small molecules, this membrane is impermeable to ions. So they will not be able to diffuse across the membrane. Not easily get across. They will be able to get across, but they will require special proteins to help them get across, which we'll talk about in just a moment. Now I want to talk a little bit more about these ions and consider some of the relative concentrations inside the cell and outside of the cell. Okay, so let's talk about ion concentration. Your book gives specific concentrations of these ions. I want you to know the relative concentration, meaning are particular ions in a higher concentration within the cell or outside of the cell. And so let's make a little table. Here we will have our ions. Here we will have inside the cell. And here we will have outside. And let's talk about these ions: sodium, potassium, magnesium, calcium. I just noticed on your note-taking guide I put C++. I certainly didn't mean doubly positive carbon. That's just typo. It's supposed to be calcium in your note-taking guide. And then chloride. Sodium has a greater concentration outside the cell, so I'm going to just give it a plus with the concentration symbol around it. And sodium inside the cell is lower concentration in comparison to outside the cell. Potassium is higher in the cell compared to outside of the cell where it is lower. Magnesium, it's a very low concentration both inside and outside, but it doesn't take a lot to do what it needs to do. Outside of the cell, it has a higher concentration. It's about 4 or 5 times higher. Inside the cell, then it would be lower. Calcium is higher outside of the cell than inside the cell. Now there are parts inside the cell, like the ER, that's going to have a very high concentration. But we're just talking about in general inside of the cell. And then the last one is chloride. Chloride has a higher concentration outside of the cell compared to a lower concentration inside the cell. So don't worry about concentrations specifically, but you should know the relative concentration, higher or lower, inside or outside of the cell. What you'll notice is that potassium is the only one of these that is in a higher concentration inside the cell compared to out. Everything else has a higher concentration outside. Maintaining these concentrations sometimes will require the cell to expend a little energy, or maybe even a lot of energy, in order to maintain this relative concentration of these ions. Now what I want to mention now are some of the proteins that are involved in membrane transport. Getting some of those molecules across the membrane that are going to have a harder time moving, like the ions or the larger molecules, may require some help from these proteins. So let's draw our membrane. And I'm not going to draw the fatty acid tails here, but you should know that they are there. The first type of protein I'm going to talk about that's involved in transportation is called a transporter. I'm going to draw it like this. Now I want to draw this molecule that is going to move through this transporter to get from this side of the cell membrane to this side of the cell membrane. So here's this solute. It binds here into an active site, or a protein binding site. It causes a conformational change of this transport protein, causing this end to open up now. And now that solute that was on this side here will now move towards this side because of that conformational change of these proteins that forces it across the other side. In doing so, you can see how this solute never had to touch this membrane. It never had to find a way to dissolve in this nonpolar environment. It was able to bypass the membrane by using these transporter proteins. And I want to write that word I said here because it's an important word when we're considering this kind of movement. Conformational change. It requires this conformational change once it binds the solute. I also want to say that transporters are fairly specific about what they're moving across the cell. Next, I want to talk about channel proteins. I'm going to extend the membrane a little bit, and let's go ahead and draw this channel protein. Using channel proteins, certain solutes can now freely pass between this channel. There is some specificity, but in general, these channel proteins are less specific and don't require a conformational change. Now that we understand the proteins that can be used to move solids across the membrane, I want to now talk about the different ways or the different mechanisms that cells will use to move solids. Let's call this board here mechanisms of transport. So let's draw our famous membrane here. So I want to make sure we talk about simple diffusion, passive transport, and active transport. We'll start with simple diffusion. When we talk about simple diffusion, we mean that molecules can move across the membrane without help, meaning proteins don't have to help move it. We say that this kind of movement moves down the concentration gradient. And what we mean by that is we're going from a low concentration here to a high concentration here. And an example of this might be oxygen. Oxygen would move across the membrane using simple diffusion. So you may have one oxygen molecule here. So over here you may have several oxygen molecules, a high concentration of oxygen, and a relatively low concentration on this side of the membrane. So it's going to go from a high concentration to a low concentration. So it's going to diffuse across the membrane. Remember, polar molecules can't do this because of this hydrophobic area of the membrane. Okay, passive transport. At one time you may have learned this as facilitated diffusion. If you were to call it facilitated diffusion, I wouldn't take off for that because it is an acceptable way to call it. But this is a situation where molecules need proteins for transportation. Like simple diffusion, passive transport moves down concentration gradient. Now, passive transport can either use our channels like so, or it could use one of these transport proteins. A good example for transporters would be insulin. So let's say insulin is in a high concentration on this side of the membrane and a low concentration over here. Insulin will bind into this transporter, conformational change occur, allowing it to be released over here until you end up with roughly equal concentrations on both sides of the membrane. Glucose is a good example for one of these channels, and it will move through the channel from a high concentration to a low concentration. Okay, now let's talk about active transport. This is a situation where molecules need proteins. So in this sense, they're like passive transport. So they need protein and energy. Passive transport and simple diffusion do not require energy. So this is the first one that requires energy. So it needs this energy for transport. We say that this moves against the gradient. You could also say it moves up the gradient, but we usually say it moves against the gradient. It will also require a transporter. Channels won't work so well with active transport, so they require these transport proteins. And so let me draw a transport protein. And good example of this would be the ions. So maybe chloride we'll talk about. And it's going to go against the gradient. So in this example here, we're going to have a low concentration of chloride on this side of the membrane and a high concentration on this side. If it was going to be by passive transport or by simple diffusion, you would expect these chlorides to go this way. But this kind of transportation goes against the gradient. So chloride's going to bind from the low concentration. It'll bind to this transporter, change the conformation, and release it on this side, making the concentration of chloride, in this case, even higher on this side. I want to label these here real quick. So I'm going to erase the bottom part here. And I want to talk a little bit more about passive transport. And I'm going to talk about some of the factors that influence how it moves. One of them I've already talked about, and that is the concentration gradient. And when we think about the concentration gradient, the only thing we're thinking about is the concentration of the solute on one side of the membrane compared to the other side of the membrane. And it's always going to go from a high concentration to a lower concentration. The other factor that drives passive transport is the electrochemical gradient. So what I'm saying with the electrochemical gradient is the amount of charged particles on one side of the gradient compared with the amount of charged molecules or charged solutes on the other side of the gradient. So we would expect them to move from a large number of positive charged solutes to a smaller number of positive charged solutes. Now I want to talk a little bit more about active transport. As I said here, it's going to move against the gradient, the concentration gradient. So it will move from a low concentration to a high concentration. These would be endergonic reactions. So just to review what we mean by that, what we're saying is that the delta G is greater than 0. So this will not be a spontaneous reaction. It will require energy. So this will require the use of ATP. So it's needed for active transport. Just like with passive transport, active transport can also take into consideration the charge. So it's not just concentration, it's also charge. So thinking about charge, this is important in nerve propagation and respiration. So I want to talk a little bit more about active transport, but I just needed a little bit more space. I'm going to talk about the energy that it's going to need to move against the gradient. So let's draw our membrane. Okay, so in this picture, our solutes are high on this end of the membrane. So we'll make purple squares to show that it's higher over here. This could be electrochemical gradient, this could be a concentration gradient. And the reason I drew this arrow here isn't because that's the movement of the molecules, it's because that's what the gradient would suggest it should do. But we're going to go against the gradient here. The first way is to use ATP Driven Pump. And the way this might work is you might have your transport proteins here, the molecule binds here, but remember it's going to require energy in order to do this. And on this transport protein there may be an ATPase or another ATPase near here. But at any rate, it's taking ATP, hydrolyzing it to produce ADP. Breaking this high energy bond creates energy to allow the movement of this solute from a low concentration or a low electrochemical standing to this higher end of the gradient. So this is the ATP driven pump. Now the next one is using a coupled pump. So we'll still have our transport protein like so, and our solute over here that's trying to get over here even though it's a higher concentration, but it can't do it. It needs some sort of energy input. So what we mean by coupled is that this transporter here will move two different solutes. This one, it's going to go against the gradient from a low concentration to a high concentration. The other solute is going to go from a high concentration, and I'm going to make this one a closed circle, to a lower concentration. So passive transport. So what happens is this molecule here that is in higher concentration on this side of the membrane binds to the transporter. It's going to go down the gradient, so it doesn't require energy. This purple square, what it's done, it's a little tricky. It binds here, and it's waiting to move across here. And as this orange one molecule binds, it changes the conformation, allowing it to move down the gradient. As it flips this transporter, it also provides an easy pathway for this purple molecule to now go against the gradient. I want to say that one more time. In coupled, we have molecules in a higher concentration on this side that are going to bind to the transporter so it can move to a lower concentration using passive transport. In the coupled pump reaction, what happens is this solute at a lower concentration is going to go against the gradient. It can't do it on its own. It requires energy. It needs help. And so what it does is it binds to this transporter. As soon as this transporter opens up to let this molecule go down the gradient, it is able to sneak across here and move to this higher concentration. This last one is called a light driven pump. And for this one, we may have a large multipass protein, and you're trying to get our good old purple solute across here. But it requires energy. And it receives this energy from the sun. The sun provides the energy for this transport molecule, transport protein, to change conformations and move this solute here, this lower concentrated purple square, across the membrane. I'm going to add one extra way to categorize these. We can think of this first one, using the ATP driven, as a direct energy use. Same thing with number 3. the light-driven reaction, direct energy use, because they're directly taking the energy either from ATP or from the sun to move a molecule against the gradient. The coupled pump, we would call this indirect energy use. It's not using any energy created, it's just moving across the membrane, sort of taking a free ride at the expense of this other molecule. Alright, let's now talk about a very specific ATPase pump involved in active transport. And I'm going to draw my membrane like this, a little simpler this time. Over here we have the extracellular space, and over here we have the cytosol. On this side of the membrane, it's going to have more positively charged solutes. It's going to have more of a positive charge compared to over here, where it's going to be more negative. With that said, it's going to be more difficult to move a positive ion from this side to this side. It's going to be easier to move a positive ion from this side of the membrane to this side of the membrane. Okay, I've set the scenario, but now let me tell you which pump we're talking about. And it is the sodium-potassium pump. And let's go ahead and draw it like so. Now this is a very important cellular pump. This is found in most animal plasma membranes. Potassium and sodium gradients required for many cell functions. This pump works by pumping sodium out of the cell and potassium into the cell. In general, what happens is we get 3 sodiums out of the cell and 2 potassiums in the cell. So it's not a 1-to-1 ratio of moving them. Okay, let's go ahead and label our concentrations of sodium. There's more sodium on the outside of the cell compared to the inside of the cell. There's also more potassium on the inside of the cell compared to potassium on the outside of the cell. If we think about the gradient here, we would predict the gradient, or the gradient would be going from outside the cell into the cell, from a high concentration to a low concentration. In addition, I'm making this arrow thick because we're also taking positive ions and moving them towards the negative area. So it's going to be a greater gradient here. Now the gradient for potassium will go this way. Here you're taking positive potassium ions and you're moving them to the positive area, so the gradient won't be quite as strong. Now remember, this is active transport, so it's going to go against the gradient. And because it's going to need energy, and I said this was an ATPase pump earlier, let me just write that here so we don't forget. Potassium is going to go against the gradient. It's going to go from this low concentration to the high concentration. So potassium will go this way, and sodium will go from this low concentration, actively using the energy from the ATPase here, to this higher concentration. Moving sodium from this side to this side is a little more efficient, or at the very least we get more sodiums moving in this direction than potassiums moving in this direction. And I said this was important for many functions, and I just want to point out one, and that is for nerve propagation, moving a signal from one neuron to the next neuron.